In August 2022, a camera resting on the floor of a trench off the coast of Japan recorded something that was not supposed to be there. A fish. Small, pale, almost translucent, so that the outline of its organs showed faintly through its skin. It moved slowly along the sediment, unhurried, the way snailfish move, head down, tail drifting behind it, feeling across the mud for something to eat. Above it, the water column stretched upward for more than 8 kilometers. Above that, sunlight. The fish had never seen any of it. The depth reading on that camera was 8,336 meters. That fish is now the deepest one ever filmed anywhere on Earth. It is also very close to being the deepest one that can ever exist. Because somewhere just below where that camera was sitting, there is a line. It is not a wall. There is nothing there to see. The water on either side of it looks exactly the same, but no fish has ever been found beneath it. And there is now a good physical reason to think that none ever will be. The strange part is what that line is made of. It is not pressure. To understand what stops fish, you first have to understand where they are. The deepest parts of the ocean have a name. They are called the Hadal zone after Hades, and they begin at 6,000 meters and continue down to nearly 11,000. That is the bottom fifth of the ocean's depth range, and almost all of it is contained in trenches. Long, narrow scars in the seafloor where one plate is sliding beneath another. There are around 27 of them. Together, they cover a tiny fraction of the seafloor, less than 2%. But they are deep out of all proportion to their size. If you took every trench on Earth and stacked their vertical range, you would be looking at a world that no human has ever properly surveyed. Inside those trenches, the pressure is difficult to describe usefully. At 8,000 meters, every square centimeter of surface is carrying about 800 kilograms. A sealed air-filled container sent down there does not leak. It collapses instantly into a shape that is no longer recognizable. So the obvious assumption is that this is what stops life. That at some depth, the pressure simply becomes too much and everything is crushed. That assumption is wrong. And it is wrong in an interesting way. Pressure only crushes things that contain gas. A submarine has air inside it. A human lung has air inside it. That air resists, and then it doesn't, and the structure fails. This is why deep diving vehicles are engineering problems. You are trying to keep a bubble of atmosphere intact under a force that very much wants to close it. An animal with no gas inside it does not have that problem. Water is nearly incompressible. If your body is essentially a bag of water, then the pressure outside and the pressure inside equalize and nothing collapses. You could take that animal to the bottom of the deepest trench and it would not be crushed because there is nothing inside it to crush. Hadal snailfish are built exactly this way. They have no swim bladder, no gas-filled organ for buoyancy at all. Their skeletons are barely mineralized, soft and incomplete. Their flesh is gelatinous. They look honestly like something that has already been damaged. They have not been damaged. That is the design. So if the fish can survive the pressure, and clearly they can, then something else must be drawing the line. The answer turns out to be happening at a scale far smaller than the fish. It is happening inside its proteins. Pressure does one thing to a living cell that has nothing to do with crushing. It interferes with shape. Every protein in a body is a long chain folded into a specific three-dimensional form. And that form is the entire point. An enzyme works because its shape fits the thing it acts on. Change the shape and it stops working. Pressure pushes on those folds, squeezing water molecules into places they should not be, and the protein begins to lose its structure. Deep-sea animals solve this with a class of molecules called Piezolytes. These are small compounds that sit in the fluid around the proteins and stabilize them, effectively holding the folds in place against the pressure trying to unfold them. The most important one in fish is called trimethylamine N oxide, TMAO. You have almost certainly encountered it without knowing. TMAO is what makes fish smell like fish once it starts to break down. It is present in shallow water species in small amounts. In deep water species, there is much more of it. And when researchers measured TMAO concentrations in fish collected across a range of depths, they found something very clean. The deeper the fish lived, the more TMAO it carried. The relationship was close to a straight line. That straight line is what created the problem. Marine fish have an ongoing difficulty that has nothing to do with depth. Seawater is saltier than their blood, considerably saltier. Seawater sits at roughly 1,000 units of osmotic concentration. A typical marine fish sits at around 350. Because of that difference, water is constantly being pulled out of the fish and into the sea, through the gills, through every permeable surface. A marine fish is slowly dehydrating for its entire life. It compensates by drinking seawater continuously and using specialized cells in its gills to pump the excess salt back out. This costs energy, and it never stops. Now, add TMAO to that situation. TMAO is an osmolyte. Putting more of it into the body raises the fish's internal concentration. At moderate depths, this is helpful in two ways at once. It protects the proteins and it narrows the gap with seawater, which means less water loss. But the line keeps going. In 2014, a research team took the measured relationship between depth and TMAO concentration and extended it downward to ask a simple question. At what depth would a fish need so much TMAO to keep its proteins working that its internal concentration would match seawater exactly? The answer they calculated was somewhere between 8,200 and 8,400 meters. At that point, the fish is in balance with the sea. And past that point, it inverts. The fish becomes saltier than the ocean around it. Water stops leaving and starts coming in. Every system the animal has evolved. The drinking, the salt-pumping gills, the entire osmotic machinery of a marine vertebrate is built to run in one direction, and it would now be running backwards. There is no known way for a bony fish to solve that. To go deeper, it needs more TMAO. But more TMAO is the thing that breaks it. The ceiling is not the ocean pressing down. The ceiling is the solution reaching the point where it becomes the problem. When that prediction was published, it was exactly that, a prediction. A number derived from a trend line, and trend lines can be wrong. Worse, there was already a record on the books that seemed to contradict it. For decades, the title of deepest fish ever recorded belonged to a cusk eel called Abyssal galathi, collected from the Puerto Rico Trench at 8,370 meters. That is below the predicted ceiling. If the record held, the theory was in trouble. But the record has a problem, and it is worth being honest about it. The specimen was taken by a trawl, a net dragged along the bottom and then hauled up through thousands of meters of water. A net that is open on the way up will catch things on the way up. There is no way to be certain. The animal was living at the depth where the net touched down, and the specimen itself was dead when it reached the surface. It is not proof of anything. It is a possibility that cannot be excluded. So the question stayed open until someone went and looked with a camera instead of a net. Over the following years, teams began systematically dropping baited landers into trenches across the Pacific, filming whatever came. Not collecting, watching. The pattern that emerged was remarkably consistent. Down to around 7,500 meters, snailfish are common. They are, in fact, often the most abundant large animal in the trench. Between 7,500 and 8,000, they thin out. Past 8,000, they become rare and the footage becomes precious. And then below roughly 8,200 to 8,400 meters across every trench, in years of accumulated footage from expeditions all over the world, nothing. No fish of any kind, not rare fish, not occasional fish, none. The fish filmed in 2022 at 8,336 meters is the deepest ever seen. It sits almost exactly on the predicted line. A prediction made from a chemistry graph and a camera on the seafloor arrived at the same number. Here is what makes this genuinely strange rather than merely interesting. The Challenger Deep at the bottom of the Mariana Trench is close to 11,000 meters. The fish ceiling is around 8,300. That leaves roughly 2.5 kilometers of ocean beneath the deepest fish that has ever existed. 2.5 vertical kilometers, deeper than most of the Alps are tall, of water and sediment and slope that no vertebrate has ever occupied. And it is not empty. Lower a baited trap to the floor of the Challenger Deep and leave it for a few hours. And when you bring it back, it will be full. Amphipods, small crustaceans, shrimp-like, pale, arriving in numbers that are difficult to believe given how little food reaches that depth. One species, Hirondelia gigas, dominates. It is found throughout the deepest parts of the Mariana Trench, right down to the bottom. Getting there required solving a problem that has nothing to do with pressure or salt. Deep, cold, high-pressure seawater dissolves calcium carbonate. That is a problem when your entire skeleton is on the outside of your body and made of calcium carbonate. At those depths, an ordinary crustacean shell would begin to break down. Hirondelia produces an acid that pulls aluminum out of the sediment it lives in. That aluminum reacts with the surrounding seawater and forms a gel over the animal's shell. A coating that resists the dissolution. The amphipod is essentially mining the seafloor and wearing it. It also eats wood, sunken plant material that drifts down from land, broken apart by enzymes that let it digest cellulose. At a depth where almost nothing arrives, it survives partly on the remains of forests. There is more down there than amphipods. Sea cucumbers moving across the sediment, polycate worms in the mud, and xenophyophores, organisms that are a single cell, but a single cell that can grow to 10 centimeters across, building an intricate structure out of sediment grains. They have been found in the Mariana Trench well past 10,000 meters. And beneath all of that, in the sediment itself, the trench floor turns out to be biologically busy. Measurements taken in the Challenger Deep found microbial activity roughly twice what was recorded on the nearby abyssal plain, 4 kilometers shallower. The reason is shape. A trench is a V. Organic material that drifts down across a huge area of ocean gets funneled along the slopes and concentrated at the bottom. Earthquakes shake more of it loose. The deepest place is not the emptiest place. It is a collection point. So why do all of them get to be there and the fish does not? Because they never had the fish's problem to begin with. Almost all marine invertebrates are osmoconformers. Their internal concentration already matches seawater. They are not fighting a gradient because for them there is no gradient. They can load their tissues with as much stabilizing chemistry as the pressure demands, and there is no crossover point waiting to invert on them. The barrier at 8,300 meters is not a barrier in the ocean. It is a barrier in one particular body plan. Bony fish inherited a specific solution to living in salt water. Keep the blood dilute, drink constantly, pump the salt back out. On the surface of the planet, that solution is spectacularly successful. There are more species of bony fish than all other vertebrates combined. It works in reefs and rivers and open ocean and under polar ice. It just happens to have a flaw. And the flaw is at 8,300 meters, and it was set hundreds of millions of years ago by an evolutionary decision no fish was aware of making. The last thing worth sitting with is how unlike a boundary this is. If you were somehow able to descend through that depth, you would notice nothing. No change in the water, no pressure threshold, no temperature shift, no visible line. It is dark above it and dark below it. A submersible passes through without an instrument registering anything at all. But it is the same depth in every trench on Earth. The Mariana, the Kermadec, the Japan Trench, Puerto Rico. Oceans that have been separated for millions of years, populated by fish that have never encountered each other. And all of them stop at the same place because they are all running the same chemistry. Below it, the trench keeps going, sometimes for 2.5 more kilometers, populated, active, feeding on wood and sediment and whatever falls from a world it has no way of perceiving. We spent a long time assuming the deep ocean was empty because it was hostile. It turns out the deep ocean is mostly full. There is just one lineage that cannot follow, and it happens to be ours.